So today, what will we be learning? In today's lesson, we'll take a look at passive and active transport and how they move molecules across the membrane. We'll look at how concentration gradients are formed, and then finally, how large molecules move in and out of the cell. So let's dive in. A concentration gradient is when a solute is more concentrated in one area than another. A membrane separates the two different concentrations of molecules, allowing for that concentration gradient to form. So that leads us to passive transport. Passive transport is the net movement of molecules from a high concentration to a low concentration without metabolic energy, such as ATP, needed. This plays a primary role in the import of materials and the export of waste. There's two types of passive transport. The first type is diffusion, where the movement of molecules from high concentration to low concentration is just directly across the membrane. What type of molecules do that? That would be small nonpolar molecules such as nitrogen, oxygen, carbon dioxide. But what if their molecules are not small or nonpolar? Well, they then move through via a process called facilitated diffusion, which is again the movement of molecules from a high concentration to a low concentration, but it's done through transport proteins. It allows for hydrophilic molecules and ions to pass through the membrane. So now let's take a look at an example. In this example, we have a plastic box with a piece of plastic in the middle with small little pores in it. And on one side of that simulated membrane are small white molecules and then larger red molecules. So let's take a look, see how passive transport works. So you'll see in the video that the small white molecules were able to get through the membrane because they were small enough and they went from their high concentration to a low concentration. The larger red molecules are still in a high concentration on one side because they were not able to get through the membrane. Now, you might be thinking, how is that passive transport when we were shaking the box to get things to move through? Well, it's just demonstrating an idea called Brownian motion, which is the random, uncontrolled movement of particles in a fluid. So what happens if the cell wants to move molecules the other direction? Well, that's where active transport comes into play. Active transport requires the direct input of energy, such as ATP, to move molecules from regions of low concentration to high concentration. So they're moving against their concentration gradient. So talking about smaller molecules, what about large molecules, such as large macromolecules? What do the cells do with those? Well, in endocytosis, the cell uses energy to take in macromolecules and other matter by forming new vesicles derived from the plasma membrane. And there's three types. The first type is phagocytosis. It's where the cell takes in a large particle, which you can see on your screen. The next one is pinocytosis, where the cell takes in extracellular fluid containing dissolved substances, as you can see on the screen. Finally, you have receptor-mediated endocytosis. This is where a receptor protein on the cell is used to capture specific target molecules to bring into the cell through a vesicle. Well, that's bringing large molecules in. Well, what happens when you want to remove large molecules? Well, in exocytosis, internal vesicles use energy to fuse with the plasma membrane and secrete these large macromolecules out of the cell. Examples: proteins such as signaling proteins, hormones, and waste are examples of things that the cells would move out through exocytosis. With that being said, let's practice what we learned today. In today's lesson, the focus is questions and methods. You're going to propose a new or next investigation based on an evaluation of the design methods. So what I would like you to do on this question is hit the pause button, read through it, and come up with your answer. Welcome back. In this question, you were looking at what could the scientists propose as a next investigation to help determine whether the organism used endocytosis to eat the bacteria. After reading through the choices, you should have come up with B as the best answer because the number of vesicles before and after would indicate an increase in endocytosis occurring, so the increase of eating more organisms and more bacteria. So what should you take away from today's lesson? Today's lesson included passive transport, which is the net movement of molecules from high concentration to low concentration without any energy. Active transport requires the direct input of energy to move molecules from regions of low concentration to high concentrations. The membrane allows for the formation of concentration gradients. In exocytosis, the cell removes large molecules through the plasma membrane. And in endocytosis, the cell uses energy to bring in these large macromolecules via the plasma membrane, forming vesicles. Well, I hope this video was informative to you. Until next time, thank you.